Greetings, everyone. Happy Wednesday, and thank you for joining us for another RRP-hosted research webinar. In fact, I actually think this might be our first for 2026. So welcome. I'm Katrina Erickson. For those of you who don't know me, I'm the Executive Director of the RUNX1 Research Program. I think typically most of you who are joining are familiar with RRP, but in case some of you are not, I always like to introduce us a little bit. So we're a nonprofit organization who is dedicated to supporting patients with RUNX1 familial platelet disorder, a genetic disease that unfortunately significantly increases the risk of hematologic malignancies, about a 50% lifetime risk. And so RRP has had one singular goal since conception, and that is to prevent hematologic malignancies in our patients and improve both their overall quality of life and their families as well. Since 2016, our work includes primarily funding research, building and supporting our patient community, and facilitating collaboration across the many stakeholders, including patients, of course, in our healthcare system, so that we can reach this goal of discovering blood cancer interceptions. Our impact in terms of research dollars has now reached over $13 million. And that's only been possible thanks to the many, many wonderful co-funding partners we've had over the years. If you're on this call and you yourself conduct RUNX1 FPD relevant research, it doesn't have to only be on RUNX1, but certainly needs to be relevant to our goal. We are interested. And if you're interested in even presenting on one of these webinars, please don't hesitate to reach out to me. I would love to hear from you. Also, if you're interested in applying to one of our grant programs, please do keep checking our website, or one of the easiest ways to keep on track of things is to subscribe to our newsletter, or you can follow us on social media. We announce our grants that way. And of course, this summer we'll have our co-funding grant opportunity for early career investigators with ALSF. That will be announced in the summer. The due date is usually always early December sometime. I don't have the exact due date handy at the moment. And then there might also be another grant mechanism that we launch later this year in the fall. So please do check in with us. And like I said, if you subscribe to our newsletter, that's probably the best way to make sure you're up to date on everything that's going on at RRP. Before I introduce our speaker today, I just want to make a quick reminder that we'll host a Q&A session at the end, hopefully around hopefully around 10 to 15 minutes. So please use your Q&A icon at the bottom for your questions, because that helps me organize the questions at the end and we can go through them one by one. But you are, of course, more than welcome to offer any comments you want during the presentation via chat. That's also fully appropriate. All right, so without further ado, I'd like to introduce Dr. James DeGregori, I hope I said that right, who is a professor of biochemistry and molecular genetics and immunology and microbiology at the University of Colorado Anschutz Medical Campus, where he also holds the Courtney C. and Lucy Patton Davis Endowed Chair in Lung Cancer Research. Dr. DeGregori's research centers on the evolutionary dynamics of cancer initiation, and specifically how aging and other environmental pressures reshape the fitness landscape of hematopoietic stem cells in ways that would promote selection and expansion of mutant clones. The work he'll be sharing with us today is particularly relevant to our patient community. His recent preprint, Hematopoietic Stem Cell Aging Promotes TET2 Clonal Hematopoiesis, describes a mechanism by which aging hematopoietic stem cells actually upregulate a RUNX1 transcriptional program. And so we're especially eager to discuss the implications of this work in the context of RUNX1 FPD. We are delighted to have you here, James, and I look forward to your presentation and to our discussion at the end. Please take it away.
Great. Thanks so much. It's a real pleasure and honor to be here. It's a wonderful organization. I went to one of the meetings I don't know, 7, 8 years ago in Santa Barbara. It was really fantastic. All right, so I'll go ahead and share. Hopefully you're seeing full screen now.
Yes, we are.
All right, so yes, so happy to be here. So I'm going to start off, as I always do, from the evolutionary perspective in terms of thinking about, why do we get cancer and other diseases, and why do we mostly get these diseases at older ages? And so we've been giving this a lot of thought over the last couple decades, and some of the modeling that we've done, and this was done by Andre Roshak when he was a postdoc in my lab, was asking to try to reconcile what to us was a bit of a paradox that you have different leukemias, for example, driven by different numbers of known mutations, CML mostly diagnosed in the chronic phase, only one mutation, and diseases like AML and CLL with multiple mutations. But it's even more striking if you look at solid cancers, with colorectal cancers being driven by 4 to 11 different driver mutations, depending on how you calculate it, down to mesothelioma, which is thought to be really just driven by one mutation. And at the same time, we know that these cancers arise in very different stem cell pools, either very few stem cells per crypt, such as in the colon, where you have about 10 per crypt, but you've got roughly 10 million of these crypts in your body, so you've got about 100 million of these stem cells, to the hematopoietic system, where you've got something on the order of 100 to 200,000 stem cells that's intermixing. So very different organization of stem cells, very different numbers of driver mutations. And if you do a simple sort of transformation where you simply graph different malignancies by their maximums, they fall right on top of each other. And to me, this is one of the most striking observations that my lab made. It's so simple yet so striking, is that this common pattern of malignancies. And what is also striking is you could put heart disease, you could put kidney disease, you could put a lot of other maladies of old age, and they also fall very similar in terms of their curves. And so I'll mention that we did mathematical modeling, and the one way we could resolve this is by having a parameter where there was differential selection with age, where in youth, there is either anywhere from very negative to close to neutral selection for different mutations. And in older ages, this converted to more positive selection. And that parameter allowed us to accommodate these different factors and come up with relatively common curves of cancer incidence. And so what we think is happening here is this is really just the manifestation of natural selection. So natural selection is pushing down on these curves for the very simple goal of delaying the cost, the fitness cost to a human of these maladies until later ages. And we think that these mechanisms that we've evolved to prevent us from getting cancer and really other diseases through most of our what would be our reproductive period are myriad. And so, everyone I think who studies any one of these different mechanisms, whether it's senescence, cell competition, inflammation, or telomere attrition, they all think that it's the bee's knees, right? That's the explanation for why we get old. Those are the explanations for why we end up getting cancer. So those are the key tumor suppressive mechanisms that we've evolved. And what I'd argue is that they're actually all quite relevant, and that we've evolved these different barriers such that in the end, fortunately, cancer is relatively rare, at least for the first half of a century of our lifetimes. And even when we do get cancer, for most of us, or for about 40% of us who do develop cancer, and that's tragic, that will come from one cell that made it past all these barriers, one clone that managed to jump all these barriers and make it to become a cancer. But the other thing I'll talk about is that these are the mechanisms that we've evolved through youth. Because remember, the currency of natural selection is reproductive output. And so natural selection is acted to maintain these barriers to the extent that's maximized reproductive output. So when you get old, a lot of these mechanisms will wane. They don't always go away, and they're still around to some extent, but they're less effective. And that's again, it's not a mechanism to get rid of us or any other animal. It's simply the waning of mechanisms to maintain us. And so if we kind of step back and think about this again in a big evolutionary picture, we can imagine that these cancer suppression mechanisms, this somatic investment, it's not just cancer suppressive, it's heart disease suppressive, so forth, right? They're mostly functional through our years when we are likely to be reproductively active. And in part, this is because, because I always get a man who will say, hey, wait a minute, I can still reproduce when I'm 70 or 80. And my answer will be, yeah, but not if you're dead. It's for if you look at historical survival, most humans did not survive past 50. And while there were some that survived past 50, their chance of passing their genes on to future generations was much less. And there are things like inclusive fitness, there's grandmother effects and so forth. And so these are certainly contributing. But overall, basically, we can imagine that natural selection has made an investment in our soma that maximized reproductive output. And for these reasons, cancer and a lot of other diseases of aging are shoved off to the right side of this curve. But at the same time, we need to understand, well, if we understand why these mechanisms are waning and how they contribute to disease, maybe we can change this calculus. Maybe we can alter some of these barriers, maybe boost some barriers or maintain some barriers. Because you could imagine if we could push these curves out another 10, 20 years or so forth, in terms of, could we really extend the health span of a human and reduce some of the burden of some of these diseases? So what we'll be talking about today is clonal hematopoiesis. And so clonal hematopoiesis is a disease, or it's not, we shouldn't say it's a disease. It's not a disease. It's a condition whereby our hematopoietic system becomes more clonal as we get older. So a fraction of our hematopoietic system will be driven, instead of one of 100,000 clones, it's driven by one clone. And that clone will often have a mutation. And that mutation will often be in leukemia, associated genes, or even mutations themselves that are associated with leukemias. And this increases exponentially as we get older. It's much rarer, before the age of 50. But by the time you're, past 70 or 80, 10, 20% of humans will have this condition. And I think what's relevant is this condition. Before I go on, this condition can be driven by a number of different cancer or leukemia driver mutations. And the most common one is DNMT3A, followed by TET2, and so forth down the line. And what I'd like to point out from this is, one, they're all highly associated with age. Some can be associated with therapy, even one's associated with smoking. But the kind of important point is they're associated with bad outcomes. And the one shown here is actually hematopoietic malignancies. But you can see that TET2 these mutations only confer a small increase in the risk of hematological malignancies. Hazard ratio just a little bit over 1. TET2 somewhat more, but splicing factor mutations confer a much greater increased risk of malignancies. And the reason I'm picking these three will become evident as we're going through. But the other important thing to realize is that this clonal hematopoiesis is associated with a lot of other maladies of old age beyond leukemias. In fact, if you have clonal hematopoiesis, you're much more likely to die of heart failure or some other cardiac problem than you are of leukemia. It's the much greater contributor to the increased risk of premature death for someone with clonal hematopoiesis. But it's associated with a number of other maladies of old age. But the other thing I find really interesting came from this study, which showed that if you map the hazard ratio for these different mutations, comparing their increased risk of peripheral artery disease, which is associated with a lot of cardiovascular diseases, and hematologic malignancy, they're relatively concordant. So, for example, splicing factor mutations substantially increase the risk of both. TET2 mutations have a more modest effect on both, and DNMT3A mutations are relatively mild on the effect of both of these. And I think that's going to become relevant later because as we think about how has evolution over millions of years sculpted landscapes that either prevent or allow these different events from taking hold within our bodies. So we know that clonal hematopoiesis is associated with both aging and inflammation. And a number of studies have shown this in mouse models. And I'm throwing these all up here because I want to acknowledge my colleagues, but if I were to talk about all these different papers, we would take the whole hour. Um, and but I think as we think about hematopoietic stem cell aging, I think we need to think of it again as being very multifactorial. It's definitely DNA damage can probably play a role, and in part that role is probably through altering epigenetic profiles, not through the actual DNA mutations themselves. And because we know that there are pretty drastic epigenetic changes in hematopoietic stem cells as we get older. And these changes are reducing the per cell functionality of those HSCs. And so you get more HSCs, for example, in an older mouse or in an older human, but any given HSC is less functional. But I think we also have to keep in mind that there are a lot of inflammatory cues. And so there's a lot more to aging than just the passage of time. It's all of our exposures. And I think we also have to realize that mice in our labs are not receiving as many of these exposures. There's a really beautiful paper from Jyoti Nangalia that was in Nature a couple years ago. I think Peter Campbell's also on it. But they showed that if they looked for clonal hematopoiesis in mice, it's really hard to find. They can find some of the mutations, but the clones are, 0.1%, not, 5, 10% of hematopoiesis. They're barely detectable. And yet, if they didn't raise these mice under sort of more natural conditions, they use pet store litter as their way of exposing these mice to different pathogens, then they actually got clonal hematopoiesis. So in some ways, we need to remember that our mouse models, by being so well controlled and clean, are lacking, I think, critical components. The other point I want to make from this slide is that these changes can be both intrinsic and sort of hardwired within the stem cells, but they can also be extrinsic in that the niche for these stem cells changes as we get older. And both of those factors are going to alter the selective pressures. So what I'll be telling you about is selection for these chip mutations. And what Marco in my lab wanted to ask was, was the selection aging dependent? And I know everyone always says, a talented. We all use the word talented before our postdocs and students were introduced. I just want to say Marco is super talented. He's really exceptional. By the way, we do a lab camping trip every summer up in the mountains just to make you all jealous. So what Marco developed is a really efficient CRISPR protocol for knocking out any gene of interest in hematopoietic stem and progenitor cells. And if anyone would like this protocol, actually it's on the preprint, but we're happy to share even more detailed protocols. But basically you can start with young or old donors. Whenever we're talking about young, we're talking about roughly 2 years old, and young will be, 2 to 4 months old. And so he'll isolate enriched stem and progenitor cells. He'll culture them and then transduce them with a CRISPR construct to deliver a guide RNA. And then he electroporates in the Cas9 messenger RNA. So it's kind of a hit and run, but it's incredibly efficient. We get, about as high as one can expect in indel efficiency, basically, because the other 30% are likely not giving an indel because they just got an in-frame deletion or change. So this is out-of-frame changes. And in the end, we also transduce some of these cells with a construct that gives a DNA double-stranded break within the ROSA 26 locus, and that'll be D-tomato labeled, and that's our internal control. And it controls for any of the effects of CRISPR. But the other critical thing is that we're using busulfan conditioning. This is a protocol that we developed that allows us to basically maintain the natural young or old environment of these recipients. It's more of a tickle than like a lethal irradiation. And this busulfan protocol basically doesn't disrupt overall bone marrow cellularity, selectively depletes the early stem and progenitor cells, and also at the same time doesn't induce much inflammation. And even that inflammation is gone by the time we transplant the cells in. So when Marco first did this with DNMT3A, he got a disappointment because we were looking for age-independent selection. And he basically gets this sort of mild selection over time, independent of age. And we could see this down in the stem cell compartment. If anything, we almost see a little bit of selection against DNMT3A in older ages. And this actually fits with the epidemiological data when they've modeled clonal hematopoiesis in humans. Overall clonal hematopoiesis, which remember is really dominated by DNMT3A, does not show differential selection across age. So we're seeing this with DNMT3A. But then he did TET2. And TET2 did show very striking aging dependence. So in these graphs, by the way, every time you see a dot, that dot is a mouse. So it's one biological time point. But what he sees in the peripheral blood is he sees increased selection over time in the old relative to young. But what I want to point out is this is across different hematopoietic progenitor subsets. But it's really most strikingly evident within the hematopoietic stem cell compartment. And what I'd like to point out is that this is normalized to the ROSA 26 control. And you see it's right at 1, meaning it's basically neutral in young and strongly selected for in old. So then he did a fitness test of these different populations. And as expected, old TET2 knockout cells have a real clear fitness advantage over old wild type cells. That's kind of what I've already been showing you. But what's interesting is old TET2 knockout cells only have a modestly increased fitness relative to young wild type cells. So it's not that these are super cells. They're just competing against crappy old cells. And old TET2 cells actually have a lower fitness than young TET2 knockout cells. Again, it's not that there's something about an old cell that makes it turns it into a super cell when it gets the mutation. It's just that it's competing against otherwise old cells. And finally, as expected, old wild type cells have a much lower fitness than young wild type cells. And we think it's really this fitness differential between old TET2 knockout and old wild type. This differential is what is accounting for this drastic selection. So then the question is, so why is TET2 knockout selectively adaptive in the old context? And so we turn to single-cell RNA-seq to address this question. And we've been collaborating really extensively with Shang Li's lab at USC, absolutely brilliant computational biology lab. And so when we did single-cell RNA sequencing, it really revealed, this restoration of youthful signatures in the TET knockout cells. So just to show this to you, and these are the hematopoietic stem and progenitor populations. You could see this ribosomal protein gene signature that's really up relative to young in this compartment with age, and it's pretty much restored or reversed by the TET2 knockout. And indeed, what's really interesting is if we take all genes upregulated with aging, they're here in orange, they're all uniformly down if you compare TET2 knockout versus wild type. So TET2 knockout is really reversing most of the increases that are observed with aging. If we take the rest of the genes that are not normally upregulated with aging, we don't see this effect. And so then we look at pathways, and what we find, and of course this is the reason I'm giving this talk here, is one of the top pathways, in fact the top pathway we see upregulated is the RUNX1 pathway. And it's up by middle age and old age in hematopoietic stem and progenitor cells, or hematopoietic stem cells within mice, wild type mice. And we also see upregulated the p53 signature in the same. But what's critical is in both cases, these are then downregulated by TET2 knockout. Keep in mind the 0 is the dotted line. So they go up with old age, and that's reversed by the TET2 knockout. We see the same thing for some other pathways that are up with old age as well. And this is where work from Nancy Speck's lab, who is, of course, highly involved. Sorry, Nancy, I forget your leadership position within the RUNX1 group right now. But I've been a major player within your organization for a long period of time. And what Nancy's lab had shown before was that RUNX1 naturally regulates a program of these ribosomal protein genes. And you get this uniform increase in a bunch of these different ribosomal protein genes during stress. And then if you lose RUNX1, it prevents that upregulation and prevents that stress response. And so indeed, we see very strong enrichment for these same RPLs and RPSs, these ribosomal associated proteins. And the other previous work is from the Suzuki lab. And what they had shown is that if they look by in hematopoietic early progenitor cells for binding of RUNX1 and TET2 to different DNA binding sites, that there's about 80% overlap of these sites. So in other words, that there's a lot of the RUNX1 binding sites are actually co-occupied by TET2 or TET3. So in this plus previous work that has shown that excesses in ribosomal proteins can lead to inhibition of MDM2 and thus activation of PP3, we have a model whereby with aging, you're getting increases at this TET2 RUNX1 mediated upregulation of these ribosomal proteins. And well, I'll note that we looked at the ribosomal RNAs, that are basically the scaffold, and those are actually down in old age. And that's consistent with some work by Manu Pasage's lab from a while ago. So what we would really expect is a significant excess of these proteins, and this excess is going to lead to p53 activation. And I'll mention that I don't have time to go into it because I want to show you some other stuff, but if you go to the preprint, we've also shown that knocking out either RUNX1 or p53 is also adaptive in old hematopoietic stem cells. And we've just recently shown in response to reviewers, actually really good suggestion from reviewers can be good, that if we co-disrupt RUNX1 or p53 with TET2, it actually doesn't, we at first thought, oh, that might prevent TET2 from being selected for, but it actually leads to even greater selection. And that indicates that these knockouts this is not a simple linear pathway. In other words, perhaps we shouldn't be surprised that p53 is doing a lot more than just be regulated by ribosomal proteins. It's playing other roles within, leukemia development. And in fact, p53 and RUNX1 are frequently co-mutated with TET2 in hematologic malignancies. So we then want to ask, well, is this the case in humans? And so we collaborated with Stephanie Shee's lab, and they have done previous work looking in human hematopoietic stem and progenitor cells. And so the first thing, if we just look in human bone marrow HSCs, looking either at middle age versus young or old versus young, we see increases in all these same pathways. Ribosomal proteins, RUNX1 programs, the RPGs, these are the RUNX1 regulated ribosomal proteins only, and also p53. Oh, p53 specifically regulated RPGs, the ones that actually regulate p53, I should say. And then if we look in, she's also looked at different categories of hematopoietic stem cells, and there's a more inflammatory component, and we see upregulation within that compartment as well, but not within the HSCs that seem to be maintaining a more youthful state. But finally, and perhaps most importantly, she looked at clonal hematopoiesis driven by TET2, and within the same person, she could look at the wild type cells, wild type hematopoietic stem and progenitor cells. And you could see that TET2 is downregulating all these same programs. So this is really nice because basically it shows what we're showing is not just a mouse thing. This is actually present within the humans. And finally, I'll point out that p53 upregulation in old age and old HSCs is seen pretty much across the board for these different in these different studies. So it's very common to see this. And it's also interesting, the HSC1 and HSC2 are basically the equivalent of these. It's observed within the HSC-IM-like cells, but not within the other cells. So to summarize what I've shown you so far is old age is selecting for TET2 loss in hematopoietic stem and progenitor cells, and this is mostly within the HSCs. And this expansion of the TET2 knockout cells in old mice is due to the reduced fitness of the old wild type cells. The other thing is that ribosomal, RUNX1 target, and p53 target genes are all increased in HSCs. And the loss of RUNX1, and that's all in can be reversed by TET2 loss. And the loss of RUNX1 or p53 restores the fitness of old HSCs, suppressing TET2 mutant clonal expansions. And this is available on Research Square if anyone wants to check it out. So the other thing I want to point out is that when we've done these experiments, it appears that this is cell intrinsic for the HSCs, this test2 knockout effect, in the sense that the age of the recipient mice actually doesn't matter. All that matters is the ages of the hematopoiesis, meaning the other hematopoietic stem cells that are competing, for example. So it's not exactly cell intrinsic, but it's hematopoiesis intrinsic. But we think that this is probably related to lifelong exposure to inflammatory cues and other stressors, alters these HSCs epigenetically, reducing their fitness, so that many of these changes essentially become hardwired. And it's also notable that TET2 mutant cells, particularly the mature ones, can actually generate a hyperinflammatory environment, and it can exacerbate this fitness differential. So now we're going to turn to the third mutation, which is UTF1. And if you remember, it was one of the ones, like other splicing factor mutations, that confers a much greater risk of hematologic malignancy, but also for cardiovascular disease as well. So we want to know, well, are there differences in, sort of how organisms have evolved mechanisms to prevent these expansions from happening, particularly during youth? So just as a reminder, UTF1 is critical for recognizing the 3' splice site, and this also plays a pretty significant role in differential splicing. And in fact, mutations of this have been shown in both MDS and AML to alter splicing program, increasing intron or exon inclusions and exclusions. So when we started using these models, at first we got kind of a surprising result because we started out with young mice. And when we looked in the young mice, when we basically put the mutant cells into a young environment using some of the same protocols I've shown you with the busulfan conditioning, we really get a striking selection against the mutation. So, and this is a log2 fold chain. So really the mutant cells are really just being eliminated from these mice. And so we thought, well, that's interesting because that doesn't sound very much like an oncogene, right? If it's greatly reducing the fitness of the cells. But we, meaning Bridget. Oh, I should mention Bridget. This is all being done by Bridget Hogue. She, and I use the super talented moniker for Marco, and Bridget completely deserves this too. Absolutely super student. And so what Bridget also did is she did the cross. So she took young stem cells, put them into old mice, in addition to putting them into young mice, and old stem cells went into either young or old. And that's where we really saw something interesting. So first, I'll just go straight to the right. And so while we get strong selection against these S34F mutant cells within young, we actually get positive selection for these same cells in old. And this is within HSCs, long-term HSCs. And what you could see is the in-betweens show exactly that, in between. So if we take young stem cells and put them into old stem cells and put them into young mice, or young stem cells and put them into an old mouse, we see we don't get nearly as much negative selection, but we also don't get the positive selection. So sort of indicating that they're both hematopoiesis intrinsic and extrinsic influences of the host that are influencing this selection. So when we looked so what I just showed you was in males, and indeed this is just showing you again, we get this positive selection in old age in males. It's contrasting with the negative selection in youth. And we also see this for another mutation, although we never see quite as much negative selection in youth with the Q mutation. That's more variable. But what was really striking is when we looked in females, we no longer get this positive selection in old age. And this is interesting because splicing factor mutations in hematopoietic malignancies like MDS are highly biased towards males, upwards of 80+%. So this is suggesting that there's real differences in selection at the level of hematopoietic stem cells that might account for this, the fact that the predisposition for these mutations within malignancies is so heavily male biased. So another factor was that UTF1 mutation leads to HSC accumulation. So we can see this with the blue here. So you can see that particularly in the old environment, having the mutation increases the fraction of hematopoietic, the progenitor pool that is in the HSC compartment, suggesting that it's preventing differentiation. And indeed, when we look at selection across different early stem and progenitor populations, again, we see this positive selection within long-term HSCs in the old specifically, while negative selection in the young. In the other compartments, while we still observe the negative selection, we no longer see this positive selection. So the positive selection is really completely restricted to long-term stem cells. And that's really the population that matters, I should say. And if we look at more mature compartments, we again don't see any positive selection in old age, which are the first two bars, but we see very consistent negative selection across all these within the young compartment across the board. So then again, we want, well, what's the mechanism? How do you get negative selection from the same mutation within the same pool in young, converting to positive selection in old age? And obviously, this could contribute to the fact that these are highly associated with old age. You really don't see clonal hematopoiesis driven by splicing factor mutations in younger people, at least not normally. And so we did scRNA-seq on these HSCs, and now we're collaborating with Austin and Stephen, a postdoc in his lab. And here I'll note that everything I'm going to show you is specifically within the HSC pool. We've looked at these other compartments, but I don't have time to go into those. And when we looked at these different patterns, we really got striking results. So that these patterns could be very differential. So if we just compare old mice to young mice, we get upregulation of the p53 program, inflammatory programs, and downregulation of proliferation programs. But if then we ask, well, what's the effect of the mutation? In old, we're actually getting even further upregulation of these programs because now this is compared to old wild type. But in young, we do get the opposite. We get downregulation of these inflammatory programs and upregulation of the proliferation programs, and the exact opposite. And you might think, well, wouldn't that mean that this should have a greater fitness effect in young, more proliferation, less inflammation? But what I think is happening here is that the mutation is enhancing these aging-associated increases in inflammatory protein pathways. But we really were wondering, well, could the mutation be basically resistant to these inhibitory paths of inflammation? So it's almost like it's creating a poison that it itself is resistant to. And I'll note that this is using both progeny pathways and hallmark pathways, but I just kind of pointed it out in the progeny pathways. If you ever tried out progeny, I highly recommend it. They've really come up with a tool that specifically looks at the impact, the expected impact of the changes, not just sort of whole pathways where changes could go in either direction, basically. So what we notice, though, we see that UTF1 is increasing these inflammatory pathways if you compare the mutant to the wild type old. But if we then ask, what about like fitness genes for stem cells? We see increases in all the quiescence pathways, but we see decreases in cell exhaustion pathways. And at the same time, as I already showed you, increases in inflammatory pathways. And then if we look in young, we get exactly the opposite. I find this just fascinating. Same mutation, only difference is age. We're getting exact opposite pathways. You know, it's not 100% clean. You get some variability, but particularly this cell exhaustion pathway is really up in the young, but down in the old. So we think that basically we're better maintaining a stemness phenotype, a quiescence phenotype within these HSCs, despite the fact that they're inflammatory. Because we know from previous studies from my lab and other labs that inflammation is known to cause exit of cells from stemness and reduce their stem cell partition, increasing a differentiation. And yet we get quite the opposite in the young. And so if we look at these stress response pathways that are upregulated in the young, like the UPR and the G2M checkpoint, we could see that in young stem cells, again, upregulation of proliferation pathways, upregulation of cell stress markers, and upregulation of exhaustion markers. So this kind of helps explain why you're getting the opposite fitness effects depending on age. So what was interesting is now when we started looking at long read sequencing, so we've done long read single cell sequencing, and I'll really hats off to Austin's lab. They've really technically worked this out. This is a very difficult protocol, but we get some differences that are really fascinating. And all these data are within a couple weeks old, so we have not validated any of this, just to head off any questions on that. And in fact, if any of you end up being reviewers of this paper, I'll note that I don't think any one of these changes is going to explain the full phenotype. I think we can make stories from these changes and they fit the phenotypes. But I think obviously these are programs. These are not single gene changes that are going to make the difference. But REST is a transcription factor that's been shown to be important for preventing exhaustion of HSCs. And it's the full-length REST that prevents the HSC exhaustion, and the shorter one is thought to be more dominant negative. And what you see is in old HSCs, we get more of the long functional form and less of the shorter form that tends to go up with old age in the wild type HSCs, really providing a good explanation for why this mutation in the old HSC is going to promote a stem cell phenotype, prevent exhaustion, basically. As another example, there's a small nuclear RNA that's known to promote self-renewal and prevent differentiation by sequestering certain microRNAs and modulating inflammation. And what I'll note is that is the short form, which is more dominant negative, is high in the old wild types and it's low with the mutation. So it reverses that. And it's quite the opposite in young, where we get more of the splicing of the sort of the more dominant negative one. How it is that the same splicing factor mutation can cause opposite effects on the same gene depending on age, I would love to know this. And we'll probably spend the next 5 years trying to figure that out. But what you notice if we break these down into young versus old, so you're getting, again, increases in the more dominant negative form in the young HSCs, but decreases in the old. And actually opposite if we look at the long form, where we're getting more of it, not a lot more of it, but more of it in the old mutant HSCs. So again, the speculation is that this mutation may selectively confer on old HSCs greater resistance to differentiation signals. So to summarize this, the U2F1 mutation is differentially adaptive in young and old HSC pools. And in the old HSPCs, the U2F1 S34F promotes an inflammatory transcriptional program, but we think the mutant HSPCs are resistant to this negative impacts, the negative impacts. And indeed, in the old stem cells, the mutation is enhancing stem cell quiescence, conferring a selective advantage over exhausted wild-type HSCs. And in the young HSCs, the same mutation is inducing a heightened stress response that drives aberrant proliferation and stem cell exhaustion. And finally, we've shown that this mutation leads to altered splicing in HSCs in an aging-dependent fashion that likely contributes to this differential fitness impacts. So if we step back and we think about this from, again, this perspective of natural selection, we basically can think about how this happened over evolutionary time. So you can imagine a mutation that is basically negative. Its expansion within the soma would cause premature death, for example. And we can also imagine that other mutations, maybe they may require two mutations, would, if they accumulated and expanded, would cause a much later death. So the fitness impact of the red mutation is much greater than the blue or yellow mutations. So thus you can imagine that there's going to be greater natural selection against the red mutations to prevent these expansions through youth, and less selection in the blue and yellows. And this would lead to this common curve in malignancies driven by these different mutations, which is what we see. Different malignancies driven by different types of mutations all seem to have the common curves. This is the function of natural selection acting through changing the germline, through changing, our makeup to prevent expansion of clones that could reduce our fitness. Our fitness or that of any animal, really. And so if we go back to our three, sort of mutations here, if we think about DNMT3A going through the splicing factor mutations, the risk of disease is much greater if you have an expansion driven by the ones on the right. But if we look at the prevalence within the population, it's the opposite. So essentially, natural selection hasn't worked as hard to prevent the DNMT3A mutations from expanding. And in fact, if we think about negative selection in youth, there's no negative selection against the DNMT3A mutations. In fact, they're even somewhat positively selected in youth. And the TET2 mutations are kind of in the middle on all of these. And the splicing factor mutations, there's basically natural selection has sort of done a hell no, you don't. You are not going to expand when youth because these are the mutations that would have the greatest effect on disease and thus the fitness of the organism. And again, if we step back and think about this from the perspective of hematopoiesis, we need to think about how this lifetime of exposures and even our underlying genetics is altering these selective dynamics. And I did have some stuff that I was just going to present just in case, but I'm going to skip it because I want to make sure we have time for questions. But I'll mention that we're looking at transcriptional heterogeneity, and we've been looking at transcriptional heterogeneity in hematopoietic stem cell pools because we're very interested in sort of how it is that aging is causing this sort of loss of normal functional identity. And also how that increased heterogeneity could serve as sort of a reservoir, an increased phenotypic heterogeneity that could allow selection in older ages. And I'm going to just show you one quick result. So we've used cell tagging, again, great collaboration with Shang Li, and led in my lab by the super talented Marco. We've used clonal tagging on these HSCs. And what we can basically see is that with age, we're getting increases in heterogeneity because correlation means lower down means less, more heterogeneity, less correlation. And it's both intra and interclonal, meaning even the rate of generation is greater. But what I want to show you is we did this cross where we took the same young or old donors and put them into young or old recipients. And when we analyze sister clones, so same clone in young, going from a young into a young or old, or going from an old donor into young or old. So if it's going across, it's the same clone. You can see that there are clones that are not maintaining stem cells when they go into a young background, but do maintain them in an old background and so forth. So the clone is being basically, its behavior is dictated by where it ends up. And we did this systematically. I just want to point out that when an old clone goes into an old background, it's much more adaptive than when it goes into a young background. So we see greater self-renewal numbers of clones when old goes into old than when old goes into young. And the exact opposite in young. Young self-renews better. More clones have better self-renewal when it goes into young than old. And the same thing when we look at clonal fitness. That's the size of the HSC clone. You can see much greater clonal fitness increases in old into old, than old into young. So basically, these stem cells, by being in an old mouse throughout a lifetime, have basically evolved and adapted to be best adapted to an old environment. And we could see this with this sort of ability to adapt to the environment. And I'll skip that. I'm going to skip the rest of it. And I should mention this heterogeneity is not random. It's driven by specific transcriptional programs. And we see this heterogeneity in humans as well. You can see it here, this increased heterogeneity in the old humans and the old mice. In fact, it's even greater than what we observe in mice. And perhaps that's not surprising. And I'll mention that we see pretty good overlap between this differentially variable genes between the human and the mouse cohorts. And so this increased heterogeneity, I think, is actually providing sort of fodder for adaptability. And so in some ways, we could think about aging not just as sort of this continuous reduction of fitness, but this continuous adaptation to a changing niche. And that was incredibly fast. But if you want all this, I'm pretty sure we're going to submit this week, and we'll preprint it. So within a week or so, it should be pre-printed, and you'll have access to it. So just to thank everyone again, I really super proud of my lab, fantastic lab written here, and wonderful collaborators. I just really want a real shout out to Shang Li's lab at USC and Stephanie Shee's lab at Prince Margaret. All the artwork I showed was done by my son. He's fantastic artist, and he also did all the artwork for my book. So with that, happy to take any questions. And that's our lab motor, lab kind of picture, us fighting the crab.
That's great. Thank you so much, James, for a beautiful presentation and lots to digest and think about. I feel like we need to have you come back and you had a lot of slides there that you weren't able to show. We already have a question, and those of you who are online, please don't hesitate to ask any question. All questions are good. So the first question is, thank you for sharing your recent work. Your pathway analysis results show a marked MYC signature, a
Mm.
factor that also regulates ribosomal biogenesis and transformation. What are your thoughts on its possible implication on chip selection and in the context, of course, of RUNX1, TET2, and TP53?
Yeah, no, we're very interested in that mixed signature. And the short answer is we haven't been able to follow up on it. And it's because, it's easy and the methodology for inhibiting, getting rid of a gene is pretty straightforward. We could CRISPR anything. And so we can look at PP3, we can look at RUNX1. MYC is much tougher. And we also know that MYC would be a tougher one because not only do you have to regulate it, but you have to regulate it very carefully because too much MYC's a bad thing too. So we're totally with you. We know that Mick is regulating these, and we haven't figured out the best way to study it, is the short answer.
Hmm. Well, thank you. I was especially struck again by the overlap between TET2 and RUNX1. And
Yeah.
maybe I missed this, but were you able to show that TET2 is directly demethylating the promoters or enhancers of RUNX1 target genes? Uh,
Yeah, we have not. I mean, part of the problem, of course, is this seems to be really restricted to the HSCs, and the same thing was true with the splicing factor study. And that's cool, but it also makes our life very difficult. It's very difficult to do the biochemistry.
Mhm.
So I have to look back and see what's been published on that, whether it's been shown that, I mean, that is sort of like what we're inferring, but
Right.
I don't know if it's been specifically shown for the RUNX1 target genes, whether you're seeing when you lose TET2, do you get demethylation, or no, you would get restored methylation of those
We get
programs
restored methylation.
of
Yeah.
those sites. I'm willing to. I bet there's something out there. I bet if we mine data, it's a great question. We
Yeah.
should mine that.
I think it's especially interesting because when we look at the
Yeah,
clinical data that's come out of the natural history study led by Paul Liu, what they see is TET2 mutations happen to be the second most common
yeah,
mutation in clonal hematopoiesis, in individuals' clonal hematopoiesis. And then it happens to be the most common at this point somatic mutation in those with hematologic malignancies. And I thought it was really interesting that you said it looks like it's not that there's a complete redundancy, there's
yeah,
additive effect.
yeah,
And it'd be really interesting to understand what are the differential genes there that are being affected or
yeah. No, exactly. I mean, we. It was actually in response to a reviewer. We had tried it before anyway, but we got an experiment going, and we're just getting the results where we if you mutate TET2, the question is if you add the mutation of RUNX1 on, is RUNX1 no longer selected for? Well, because you've already got TET2 loss. And the answer is it is because it's doing other things as well. So we see basically a combinatorial selection. So the TET2 RUNX clone is more fit than either individual clone by itself. So
Hmm.
there is a further selection for RUNX1 loss, and there's a further selection for PV3 loss. But if we look at hematologic malignancies, this fits, right? Because they
Hmm.
are co-mutated. I think more than one would expect by chance, but I'd have to double check on that.
Yeah, I think in sporadic. I don't know, and maybe if
Yes,
there.
and sporadic. I don't know within the RUNX1 the
Yeah, I mean
inherited.
tattoo for sure, but I don't know about RUNX1 and P53 in the RUNX1.
But you said in individuals that RUNX1 people that inherit the mutation do see TET2 clonal hematopoiesis.
Yes.
So that fits going in that direction as well, right? So I'm not sure one needs to come before the other. I'm not sure there's necessarily an order
If
to
there's a
these.
sequence or order to
Yeah,
it, yeah, which is another interesting question. Yeah.
yeah.
Um, there was another question or comment was, it is the most common somatic mutation in RUNX1 FPD malignancies.
Uh,
Do you have any thoughts um, on this? But I think
yeah, no, I mean it's interesting is, that there's two possibilities. One is that, it's because they do completely different things. But the other possibility is that because TET2 mutations can often be heterozygous, and maybe the RUNX1 mutation isn't a complete loss of function either. So when we're doing
right.
experimentally, we're doing CRISPR, and that probably takes away
Complete
both
knockout.
copies.
Yep.
So
Yep.
I think that it could be that part of the effect is a combinatorial effect on the same targets. that it's
Mhm.
that you're doing even better at preventing these RPGs from coming up in old age.
Mhm. I think,
But I think another thing is that there's different targets as well. And I'm always a believer in complexity, and I'm never a believer in simple explanations. And I think this is a case of where I think it's probably all of the above.
yeah, yeah. I was particularly struck by your model at the end where you were showing that the forces of natural selection may be stronger for more aggressive, if you will,
Yeah,
clones like UTF-1. And again, I'm always bringing it back to RUNX1 FPD, so forgive me.
yeah,
But as we look, the first somatic mutation that appears to arise is a B core, and there's a
mm,
lot of question marks about whether that actually is even maladaptive. It appears
yeah,
thinking is that it's adaptive, and you don't see it as frequently in the hematologic malignancy space. Then the second is TET2, and then
yeah,
the most common sort of late event seems to be potentially RUNX1, like biallelic loss.
yeah,
And it just raises the question that also over time, as individuals age, the niche, even though in a RUNX1 setting, when you have loss of RUNX1, you have this enhanced inflammatory
yeah,
niche already at a young age. Does with aging, do you get sort of an additive effect that
right.
allows for this
Yeah,
adapt
it's still
adaptation?
tapping into the natural selection of youthfulness in terms of preventing the cancer. I mean, you could even see this with, like people that inherit BRCA mutations, right?
Mhm.
They do get, increases in cancer, but they're still mostly happening in what, from an evolutionary perspective, would be an older age. I know, we
Right,
don't consider 40 old, but
right.
for most of our evolutionary history, 40 was, was
What's
we were
old?
very lucky to make it to 40.
Right.
And so I think that, and so I think that, yeah, even people that inherit mutations and increased predispositions are probably still benefiting from the forces of natural selection that have lessened the risk of diseases through youth.
Yeah.
But I think it's interesting what you're saying about like even the different mutations, the secondary mutations that people with RUNX1 develop. Is there sort of a gradient where the ones that you do want to have, or maybe that you don't mind having, are the ones that happen early, and
First,
the
yeah.
real bad ones are the ones happening late.
Yeah.
And I think there's a lot of examples out there. I think it needs to be quantified, but even in solid tissues, and again, I don't think people have looked at it systematically enough, but if we look across ages, we see more Notch1 mutations expanding in like 30, 40-year-olds. And the P53 mutations don't expand until you're 65, 70.
Hmm.
And the Notch1 ones seem to, if anything, be disease preventing. They
Interesting.
prevent the cancers. I mean, so they're the ones you want. And the P53 ones are the bad boys. And natural selections push those out to the right.
And
And of
we
course, if we do things like if we smoke, if we do things that disrupt these balances, we're kind of screwing up natural selections, all that work that natural selection has put into this. And then, of course, there's the bad luck phenomena, whether, I mean, I don't want to say, that, if you lead the perfect life, you're never going to get cancer. We know everyone's got examples of someone who was, ate a great diet, exercised, didn't smoke, and they still got lung cancer.
right, right.
There's
Yeah.
always a component of
There's
bad luck, whether
yeah.
it's the genetic hand that we're dealt or whether it's, again, just bad luck during our life.
Yeah. There's one final really interesting question. I realize do you have an extra minute or two?
Oh, and I'm fine.
Okay.
Yeah.
Okay. So this is really interesting because we have an ongoing stem cell harvesting and banking protocol for
Mm,
patients, more because we were thinking of a gene editing strategy in the
yeah.
future. We're not there yet. But the other concept that had come up with this was whether or not you could use young hematopoietic stem cells that hopefully are somatic mutation devoid and maybe do a reset later in life
Mm,
should a RUNX1 individual not have access to any therapy yet.
yeah.
That be an approach. The question is also asking about even potentially banking young
Yeah.
cord blood. But in light of your data, as we age, those stem cells just might not be fit enough to survive an aged environment. Is that how you
Yeah. No,
think about this?
I used to always say that we should be banking cord blood, to give it to ourselves late in life, but our data really do show that you can't change the environment. So I, I think it's a real question. I think what we'd want to do is we'd want to see what are the things during life that could better maintain that environment. And
Mhm.
you and I were talking earlier about exercise, and I
Yeah.
think exercise is one of those things. But in the end, I think, I think everything we do is going to change that trajectory somewhat.
Mhm.
But I'm not a believer that we're going to, massively change it. I do not see humans living past 120 anytime soon.
Yeah.
I think we're fighting the forces of natural selection, and natural selection has come up with such a multi-parameter way of maintaining us through youth and kind of letting us go to pot in old age that I think that changing that is going to be very difficult. And I don't honestly, I think just ethically that shouldn't even be our goal. It really should be all about health span, not lifespan.
Yeah. Yeah. On that note, we should probably close up. I love that concept of health span over lifespan.
Yeah.
Thank you so much for your time. Really appreciate it, James. And thanks everyone for joining and for the questions. And yeah, reach out if you have any other further questions. Happy to reach out to you, James, if we get more
Yeah.
questions.
And I'll sign up for your newsletter.
Great. Love it. All
All
right.
right. Thanks so much. And thanks
Bye
everyone for listening. Bye
bye.
bye.
Thank you.